So, we are now pleased to welcome Dr. Sangram Bagh. Dr. Bagh is a distinguished researcher working as an associate professor in the Biophysics and Structural Genomics Division at the Saha Institute of Nuclear Physics. He obtained his PhD from the University of Toronto and then went on to do his postdoctoral research at MIT Cambridge in the Chemical Engineering Department. He then worked as an assistant professor at the Presidency University, after which he moved on to his current position at the Saha Institute of Nuclear Physics. He was also awarded the Ramanujan Fellowship in the year 2015. Dr. Bagh's lab works mainly in the synthetic bio field with a focus in three different areas, which include cellular robotics and cancer gene therapy, space synthetic biology, and quantitative reconstruction of the native gene network. And today, he will be delivering an interesting talk on programming living cells with engineering language. Synthetic bio has been the core of AIIM, and with an expert in the house, I hope you all enjoy his talk and gather some interesting information. I would like to remind all the participants once again that you can leave your questions in the Q&A box, or you can either ask them at the end of the talk. Over to you, sir.
Uh, Thank you very much. I'd like to thank you, the organizer, specifically Professor Raju Mukherjee and Tejas for inviting me. And it's really an honor to be here among so much like talented young synthetic biologists. So let me share the screen fast and let me share. Can you hear me?
Yes, sir, we can hear you.
And can you see my screen?
Yes, we can see your screen.
Yeah, so let's start this. So we have started our lab like 6 years back in Science of Nuclear Physics, and till then we're working on the synthetic biology. As all of you are synthetic biologists in one way or another, because you are the participant of the IGEM team and you are making the India visible in the world. So I don't think that we need a lot of introduction, but the kind of the introduction I'd like to tell you, and that is my own introduction. There's the question which actually brought me into the synthetic biology in the PhD, which I have studied in 2006 at University of Toronto with David Macmillan. And that time the biology is just starting, but the questions they're raising is a very important one. And I believe that the next 50 or 100 years will take to answer these questions. So one simple way people talk about synthetic biology is the engineering, right? So what is engineering? I mean, when if you ask, oh, can you hear? Hello.
Yes, sir, we can hear. We can hear you. We can.
Oh, okay, sorry. Yeah. Okay. So
Sir, could you
when
also turn your video if it's possible?
Okay, let me see. Let me see. Okay. So it says that you cannot start your video because it is host stop something. Yeah.
Okay, sir, please continue.
Okay. Yeah. So this is the example of the engineering you always can see, like this beautiful building in the Singapore, or you can see this rover on the Mars. But I can assure you, people don't generally think about engineering like this, right? So the basic question, or a very fundamental question, is that at this technological era, when you can actually control the movement of the rover on the Mars with almost a mathematical precision from the Earth, why we cannot make a Pegasus or a Hawk? Now, it is not, now if we do a thought experiment, like you want to think about the making a Pegasus, but if you start thinking about, is it possible, I mean, which genes need to be changed in order to get the wings on the horse, you cannot even think to start, where to start. So again, so the question is that, again, that when you can do this kind of engineering, why we cannot do the engineering on the biological system? So if the answer, there's a very simple answer, maybe a one-liner. So for engineering example, like this building or this rover on the Mars, you have always a very beautiful design principle, and based on which you fabricate it, you test it, you rebuild it, and you do whatever you can. When you come to the biological system, we do not know what is the design principle of the life, and that makes us harder that where to start to make a Pegasus or a hawk. So the question is that why this design principle of the life is still unknown in this technological era. I think the answer lies in the complexity of the biological system. Some complexity you have already seen in our previous talk. There's a wonderful talk by Professor Ambika, and we know that the biological system, even simple virus and cell interaction, is so complex, so high dimensional. Now, in the last century, people thought that any biological function depends on a single gene or protein. That's why we looked for a gene or a defection protein for disease, right? But the problem is when it comes to the cancer, we never find a single biological marker for the cancer. And one of the way that people actually did that, the whole whole genome sequencing of the human genome sequencing, the reason was that if you could find a gene which is can tell about the cancer, then you can actually cure the cancer. But in the post-genomic era, we do know that we cannot do that. The reason is that any biological sample, any biological function does not depend on a single gene or protein, but it depends on the interactions of them, and that it gives a very complex network. So if you want to see a glimpse of the complexity, here, where my cursors are, you can see the network of the computers throughout the world, which are connected to the internet at the year of 2009. And this right-hand side, here is a simplified representation of a biomolecular network of a bacteria. Now, if you just look at them, you can see that the complexity are actually similar. But on the top of that, the biological system is much more complex because the biological function is more dynamical, and it has, these interactions are nonlinear. They have a lot of feedbacks, and there's a noise. So at this moment, there is no mathematical framework present to actually decipher this kind of the complex network. So it looks like this is a different understand biological system. Probably it is not. So people know that many of the people from the other fields, they actually work on the networks, and there's basically the electrical engineers or the computer engineers, the electronics network. From the end of the last century, biophysicists started understanding that there is some similarity between those electrical network as well as some biological network. So if this is true, then there is a small complementary way to understand the biological function from a systems biology perspective if we actually use that system level understanding from the network. So, and this is a kind of a start of the molecular systems biology, what we loosely define today. Now, if this philosophy is correct, then it is also possible that we can actually make some small biological network following this electrical engineering principle, put it inside a cell and actually change the function of the cell or actually develop a new function. Now, if you see the hierarchy between the biological system and a computer engineering system, you can see the protein engines, which goes to biochemical reactions, then it goes to the pathway, then goes cells and tissue. But what are this beautiful thing has been done by evolution, but we don't know the proper evolutionary principle of each of these scale. So what we can do, so we can actually take some of the principles from here, which started from a physical layer to gates to modules to computers to networks. So we take that rules and put the biological scale here, not using that evolutionary principle. In that way, we may develop some new, may not be a completely a new organism, but maybe we can actually develop some of the functions. And that is the motto of today's synthetic biology. So with this fundamental question, which always actually excites me, so I'm just, instead of giving you some examples of synthetic biology, which you all know what the synthetic biology you are going, and you are in the AIIM teams. So I'm directly going that what kind of the synthetic biology work we are doing in our lab at Science Institute of Nuclear Physics. So we look at something else, and that is the artificial neural network. So we know that this is the brain, and this brain, like the trillions of the neurons are connected with the trillions of interactions, and whatever we are thinking, whatever we are doing, starting from the Zoom webinar to somebody listening, somebody is not listening, somebody, so whatever it is all coming from the interactions of the neurons. Actually, at this moment, we really don't know that how this intelligence, the consciousness of the human being is coming from the brain, because we don't know much about their principles. We do know some of the molecular biology of it. We do know some physics of it, but we don't know how it is actually working. But the question is, whatever the small thing we know about the brain, if you take that principle and try to apply, can you do something wonderful things? And actually, we can do, right? So now that is the whole idea of the artificial neural network. So now if you take a neuron, right? And this neuron, you want to abstract this neuron function mathematically. So this neuron has of inputs, x1, x2, xn, which are combined together in a linear fashion and then processed through a nonlinear fashion and gives an output like y1, y2, ym, something like that. And you like to do the abstraction of this biological neural function is a mathematical way. You call this an artificial neuron, where you give different inputs from starting from 1 to xn and are linearly combined. And they are linearly combined based on something called weight, and represented by here W1, W2, which is nothing but telling that how much of the X1 or what is the percent of the X1 is implemented or taken by this neuron. So what you are getting, a summer function X1 multiplied by W1 plus X2, W2, so on. And this is a simple linear function. And this linear function is generally processed through a nonlinear function, and it gives a nonlinear output. So this is like abstracting a single neuron function to an artificial neuron. Now, in brain, these all neurons are connected together, and together they're developing all this intelligence, consciousness, and whatever. So now this artificial neuron, we can connect together so many artificial neurons. So this output will be taken by the another neuron or set of another neurons, and you can actually make this kind of a network. And this is called the artificial neural network. But remember, we do not know anything about the brain. So neural network is a very simplified, very simplified aspect of our brain networks, right? Now, this can do a marvelous thing. For example, we all use the Facebook, right? And in the Facebook, when you have a group photographs, that Facebook asks you that, do you want to tag this particular friend? So this from the billions of the people, Facebook actually can identify a particular face, and they do it through this artificial neural network or artificial intelligence, where this number of neurons in each of the layer, this could be actually in the Facebook, it is 60,000 and the 70,000. So you can see this an enormous level of the neural network, but we can develop a very beautiful thing to this artificial neural network. Now, how this artificial neural network has been implemented. We all know whatever the artificial neural network, either the Facebook or wherever, they are basically on the 99% case, they're through the computer, actually basically through the software, right? And however, the 1% of that, people are trying to develop the hardware for ANN. And there is a buzzword you may heard is called neuromorphic chips. This is basically material-based chip where you actually making neuronal-like circuits, and they work as a artificial neural network. And you can put it in a robot or something so that they can do autonomous tasks without any taking any instruction from other computers of the outside or the software. So people also showed that artificial neural network can be implemented as a hardware in using photonics, different photonics materials. And like 10 years back, people also showed that you can use that in vitro DNA computation to develop ANN at the hardware level. So my lab asked this question: can you actually implement this hardware, the artificial neural network in the bacteria? Can you take a single bacteria and think them as an artificial neuron, and then when a population of the bacteria can work as an artificial neural network? And that is the questions we're trying to answer in our lab. And to demonstrate that, what we took a system which is called the decoder and encoder. There's a electronic device, and people use those specifically in a lot of communication systems. So for example, when you see that the police works with the walkie-talkies, so when you actually talk with your walkie-talkie, when you're talking, the sound signal is basically changed to an electrical signal, but this electrical signal then it's encoded to only fewer bits. This fewer bits information actually going through the electromagnetic wave taken by the other receiver, and that encoded signal is first get decoded to a bigger, more bit signal, and that signal is changed to the sound signal. So the decoder and encoder system are very important for a lot of communication system, including the a lot of aviation system and so. So what we want to demonstrate, this decoder and encoder with the bacteria using that ANN. So now here is the simplest decoder. It's called 2-to-4 decoder, and here is the electronic of this electronic the system, and then this is the truth table. Now, first we did this electric design, we actually abstract in an in format where x1 and x2 is the input, and these are the 4 artificial neurons, and W11, W21, all those corresponding weights, and you can see the functions are summed up linearly, and then it is processed to an processed to a nonlinear function, and then gives 4 different the output. So that is the mapping this electronic design into the ANN here. Then we mapped that ANN figure in a real biological system. And how does that happen? So X1 and X2, now your inputs are 2 chemicals, IPTG and anidotetracycline, ATC, and these inputs are going to our engineered bacteria where the synthetic gene circuit is there. So what this synthetic gene circuit is doing, they're taking this input, they're combining with corresponding weights, and then this synthetic gene circuit is producing basically a nonlinear function and produce 4 different outputs based on 4 different fluorescence protein. So that is the idea what we want to create. And now I'll go a single example how a bacteria, we call it a bactoneuron, which is basically an artificial neuron with the bacteria, how we are developing it in a single case, and then we'll see that the others will come into the place together. So here, we like to develop an biological AND gate. That AND gate has been people developed for a long time with the synthetic gene circuit. So we use the synthetic gene circuits so that only when IPTG and ATC are present, they will produce a particular output. Either it's a fluorescence protein, Venus or EGFP here. So we actually designed and constructed this particular synthetic gene circuits. And then what we do, we characterize it. We actually try to find out what is the relation in the input IPTG and ATC along with its fluorescence protein output. So when we have this dose response curve, we use nonlinear function, which is a log sigmoid function, very general function for artificial neural network, and it looks something like that. So we fit it with it, but fitting with it doesn't give you an AND gate. Now I come to an example that how ANN actually work in software, and then I'll come that how we are using in a molecular engineering principle, something like that. Now this is a leaf, but you can think about it as a face. It could be my face, it could be your face, and whatever the face, if it is photograph, it has some values of each of the pixel, right? So what Facebook does, so you identified someone. Let's say in my face, in Facebook, it is written as the Sangram. So the Facebook knows that there is the Sangram face looks something like that. Now, when in a group meeting, for example, if this is a physical AIIM meeting and I'm giving this talk manually, in a group photo, I may be there. And then you ask Facebook ask you that Facebook actually try to find out from this group photo that who is Sangram, depending on this training set data, that is the previous things. What they do, they have just a single network, looks something like that. And what they do, they actually start changing the weight, right? Weight we defined to W1, W2 before, which is connecting with the input, right? So they change, it's not a randomly, there's a lot of different kind of mathematics are there, but what they're changing this weights for a long time, and then suddenly they get a match of this network with my face. So this is going to tell you that, okay, so this is face. So here, if you see the actual mathematics, it is so simple. It is just a log segment function for a particular neuron, and what you are changing just the weight. And when this is changing weight, and suddenly you find there is a relation between the my face from the group and the my previous face. So that you're going to find. And this way you can do just not the face recognition, disease recognition from a lot of different pictures. You can try to find out some forest fires to Amazon forest, just using those kind of the neural networks there. So exactly what we are doing synthetic biologically here, the exactly same thing, but here you are changing the weight by changing the molecular, by changing a different molecular parameters in the synthetic gene circuit in terms of different binding sites, the number of sites, where you put these sites, the transcription rate, translation rate, the interactions between the protein and the inducer, and so on, so on. So here, each of the iteration you can see, if this is an AND gate, you can see there's this AND gate looks very bad, right? It looks very bad, and then each of the iterations we are going to get, we are actually improve these weight values again and again, doing this molecular engineering principle. And then finally, we come what we call is now is match with the AND gate, right? When we got that match, similar to the face matching in the Facebook, but here we do this hardware using the molecular engineering principle. Then we do the simulation. You can see the simulations looks very good. And when again we do the experiment to test that if your simulation is work, and you can see the simulation and experiment gives a good match. So that's a good thing. We are excited that we can develop a single vectorial neuron and just changing the weight and so on in a real sense. And through that, we also develop some molecular engineering principles, because as you, all the AIIM team members, you know, if you want to develop a synthetic gene circuits like an OR gate, like a NOR gate, like an XOR gate, something, then each of the cases, your design would be different. But what ANN is giving us is a very simple design, because it has a simple equation, and which is a log segment function, which is some weight and bias. And we found that if the weight signs is positive, then you need in molecular, the manifestation of the molecular engineering, it requires an activation function. If it is a negative, you need a depressant function. So that just by knowing that, you can develop any kinds of the vector neuron. And we believe that this could be a good complementary or work or the framework for synthetic biology for the coming days. So in a similar way, we develop other vector neurons. I said like 4 vector neurons for the decoder, and we took the same pain, making this, changing this the weight through the molecular engineering. And at the end, you can see we got some good results. This is the simulation, this is experiment, simulation, experiment, simulation, experiment. All they match very well. So when we have all our vector neuron ready, we just make this artificial neural network. That's by just combining them, put this inducer in a test tube or a flask or whatever you see, and then you do the experimental results. Now, if you see the electronic truth table of this particular decoder, then you see in the output level, there is 1, 1, 1, 1 diagonally, and everywhere it is 0. So in experiment, you can also see just fluorescence, 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 fluorescence. That is the 1 values logic level. In the diagonally, everywhere it is black, so there is no expression. So we are very excited about this fact that we can use that artificial neural network to make a decoder. Now, we're going to develop something more complex, and this is the encoder. So here is a truth table of the encoder. Now the question is, here we would like to take a different approach for making this artificial neural network. In synthetic biology, when people actually use always the electronic design principle and follow that electronic design principle in the biological regime to actually make that synthetic gene circuit. But what we thought, that why we need to take the electronic design? Because we are using NN, so we forget about the electronic design. We don't want to know what is the electronic design for an encoder. We just look at this truth table. It's a very, you can see it's a complex truth table. So what we have done, we divided this truth table in 4 different smaller truth table, and each of the truth table, if you can make through an vector neuron for each of these truth table, then we can actually combine them to get this behavior. So that is an approach we would like to take without following that electric circuit. So we call it the no electronic circuit diagram is required. ANN design is possible just from the truth table. So in that way, this is the 4 truth table we divided from here, and you can really see this is the 4 vector neuron. It looks like something like that, and then we actually do again the same pain of making a weight adjustment for each of the vector neuron. We actually change this weight in such a way, and then this is the results. I don't know if you can see it properly, but it is a very busy slide. But if you can compare this microscopy picture along with this truth table, you will see they are giving exactly the same match. So this way, we are now very interested, because now you can develop an complex function without following that the electronic function. Now, similarly, we want to, so as I said, that this has to be a very general framework for making a lot of new functions. So we just go on to make something called the majority function. Mathematically, it looks like that, but simply to put in English, it is something when your most of the input is correct, true, then your output in the way is false. So here I showed that electronic design, but we don't follow that electronic design. The reason we don't follow, so as I said, so if you really want to make a majority function through synthetic gene circuit in a conventional way, then you have to make this, then you have to follow this electronic design. And here you can see this input goes to an gate, their output goes to another gate, and go on. So basically you need a cell-cell communication system to make that. So if you have a more, I'd say, complex system, then, but you don't have much of this cell-cell communication system ready at this moment. So we don't know much of them. So we cannot make those kinds of things. So that is why we don't want to use this electronic design. Instead, we use this truth table. We divide it in the 3 different truth table, make the vector neuron, do this weight adjustment, and here is the experimental results. It actually, if you compare these results with the truth table, it actually follows the truth table. So that is the powerful, that is the power, I believe, for this ANN work for the synthetic biology. Now, another aspect of these things, as you and the AIIM team, you know that when you build something, you actually ask AIIM to give you some of the previous the previous team which have to develop the circuits. So here our goal is the same. So this is the ANN vector neuron which we are developing. So there's a library of the vector neuron. So when you have a library of the vector neuron, who have already developed this decoder, encoder, and majority function, the question is, whatever the vector neurons we have, can you take those vector neuron, mix them in a different ANN, and can develop a different function? And that is what we want to demonstrate to 2 important devices, this multiplexer and the demultiplexer, the DMAS. So demultiplexer we use in our computer. For example, at this moment I'm using demultiplexer in my computer, because demultiplexer says when you give a command, it go to a printer, it may go to a Zoom webinar, it can go to a presentation, and so on. Hello, can you hear me?
Yes, sir. We can.
Yes, sir.
Okay, so yeah. So now this, so that's the demask is the thing, and the mask or multiplexer is exactly the opposite. So when you have a sound system and you want to connect the sound system with your laptop, because the sound is coming from either from the laptop, either from a projector, either from a TV, mask decide which line to give the preference, and here demask device which select which line to go disconnection. So exactly we developed this scan and the multiplexer for biological system, and here we taken the 1 to 2 demultiplexer as well as 2 to 1 multiplexer. And again, we don't use that electronic thing. We just divide this the truth table, and we find we in library we have this vector neuron, this vector neuron, this vector neuron, this vector neuron. We mix them and match them, and we find exactly the same behavior. So I'm sorry because this slides are very busy, so I cannot going through each of them. But just to get an idea, I mean, just believe me at this moment that this truth table and this is the same, this truth table and this is the same. But you can anytime you can actually look at and ask me that if it is a really correct or thing. So this is the thing. But then we also go to a different thing. Now this is about the quantum computing. So I don't know anything about quantum computing, right? But what I do know that quantum computing is some a different kind of the computational platform, and that called the reversible computing, right? And this reversible computing, it has a two property. One is has the thermodynamic reversibility, and there is the logical reversibility. So what thermodynamic reverse? So what is the use of thermodynamic reversibility is that if your system or your gate or your electronic whatever the circuit design or quantum computer circuit design is thermodynamically reversible, which means that the amount of the energy required for that function is very low because there is no energy loss. In irreversible process, there is a lot of energy loss. So you can actually develop very high efficient in terms of the energy, a very high computational power through the quantum computing. Now, what our goal is that develop the reversible computing in biological system, right? But in biological system, when we talk about a cell, right, transcription and process, or any biological process, this time I'm talking, you are listening, or whatever you're doing. So those are all irreversible. You cannot use the reversible thermodynamics, what you are studying in your curriculum, to apply in a real biological system because there's all irreversible processes. So we cannot make that thermodynamic reversibility in biological system where using the transcription and translation process, but what we can use that logical reversibility. And I'm coming in a moment, what is the logical reversibility? So here is something called Feynman gate. So this is the Professor Feynman actually proposed this. And the whole thing of the reversible logical reversibility is the information gets preserved. How? So if you see here in this Feynman gate, if you have a two input, your output looks exactly like same 0 0 0 0 0 1 goes to 0 1, but here not exactly same, but there is a crisscross pattern. So this 1 and 0 come out in the output as a 1 0 here, and this 1 1 goes here. So this information is totally preserved. So Professor Richard Feynman is one of discoverer of definitely the reversible computing. So what we found now, another question is this reversible gate, you cannot make those reversible gate using the irreversible AND gate, NOT gate, OR gate or something. So if you want to bring the reversible computing in the field of synthetic biology, you cannot follow this kind of electronic principle. So the ANN is the matter because ANN does not follow that electronic principle. So again, we take the Feynman gate, that is the that is this truth table, and divide in different vector neurons. And interestingly, all those vector neurons are already ready in our library, what we have developed for the others. So here we actually do the experiment, and you can see that it is exactly giving this reversible thing 0 0 0 0 0 1 0 1 1 0, but 1 0 is here, crisscross 1 1, it is coming 1 1 this here. So again, we are very happy that we could actually start doing the reversible computing in the synthetic biology regime in the real living cell. So with that, I like to summarize part one of my talk. And here, as I said, that we develop a single layer NN type architecture with engineered bacteria, which is able to decode and encode environmental chemical signal. And what is our main concern is that it is a general framework, single mathematical equation. We don't need a different kind of mathematical, just single mathematical equation to create any function. We develop new rules to create function without following the exact electronic design, and definitely several functions can be generated from a vector neuron library. Now we just change a gear for the next 5 or a few minutes on our second area of the research, which is the space synthetic biology. So now the space travel in this century is completely different from the last century. So the idea, if you, when the first time when people actually went to moon, the idea was to show that man can go to the moon, right? So we know as we go to the moon, we can go to the Mars. So the idea is not to go to the Mars and come back. So whole idea, if you want to make a plan to go to the Mars, the idea you want to make a basically or create a research base. If you have seen the movie Martian, they actually developed the same thing, right? Okay. Now the question is, now challenge. In the movie there is no challenge. The challenge is when you actually want to do scientifically or technologically. So if you want to go to the moon, it is like 2 years you have to go, or at least if you're the very fastest you can think, you need at least 1 year. So if you want to 2 years to go and 1 to 2 years to come back, so you have to be there at least 6 months to make that base. So it is like a 4 years, 3 years to 5 years program. And for that, you cannot take all the resources from the earth. For example, if you want to have some dosa or a biryani on the Mars, you cannot take them from Tirupati. So you have to actually cook them. Not just to cook them, you cannot take the rice, like India get the basmati rice on the Mars, because it is too costly to take all those things on the space. So what you need, you have to take whatever the Mars's resources are, whatever the carbon dioxide they have, either they have a water or whatever they have, and then you have to take those resources and make this all foods and stuff. And it is called, in technological term, is called in situ resource utilization. So what NASA feel, or many other people feel, that the synthetic biology is the key for the next generation space travel, because you are not taking anything except few vials of engineered organism, and this engineered organism will take the Mars's resources and make anything from you, starting from the road, a radiation resistant habitat, your food, your oxygen, and blah, 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 whatever. So that synthetic biology has this particular potential. And another challenge is definitely the going to the Mars, because you are going through a microgravity. So this is a radiation and microgravity in the space. And this is a 2 years journey and 2 years coming back. So it is you're going through the huge technological challenges there. Now, people know, starting from the Apollo mission, that there are 2 things happen if an astronaut goes to the space. One happened, the microgravity of the space. Due to this microgravity, there is a deregulation of the immunity of an astronaut, and at the same time, the virulence and the resistance of our own gut bacteria, which are not doing any harm in the earth, if you go to the space, they actually got virulent and actually infective. And people knew this starting from that. So people try to understand that how microgravity actually changes the lot of cellular genetic behavior at the cellular level. So people actually experimented with the human cells, bacterial cells in microgravity simulator, all in the space. But the problem is, the microgravity is very low perturbation, which basically means the change in the gene expression is 20%, 30%. And this is well below your differential gene expression cutoff, which is at least 150% to 200% required. So therefore, we don't get much data doing those kind of things. So what we develop a systems biology pipeline to analyze that low perturbation data, and we got a lot of new insights that how cell works in the microgravity. We found a lot of inflammation, cancer drug response for the human cell, and the similar to the which we published for the bacterial cells too. Now, this is not the part of today's talk. I'm not going to details here. What I just want to tell you that we got some insight from our 2 systems biology studies. That insight we used to generate some synthetic genetic circuits which works in the microgravity. So in 2015, Arkin and Adam Arkin and the group actually proposed that what are the grand challenges of the space synthetic biology. They find there is 6 different ways where the synthetic biology can make a solution there, starting from the space medicine, space cybernetics, terraforming, life support, and so on. And many of them actually require the microgravity. So what is the most important point? To make any of these things possible, you need to integrate microgravity with the biochemical processes. And there is none present at this moment before our work. So from the previous work, what we find is something called a protein called HFQ, and this is in the bacteria, not only just the E. coli, but Pseudomonas and Salmonella. In all the cases, this HFQ is get downregulated in the microgravity. And that's a good news for us, because when we develop a synthetic gene circuit with RNA, so here HFQ actually works with small regulatory RNA. They make the scaffold, bind with the mRNA and degrade it. So that is actually control the gene expression. So this is the bacteria. So now what we think, okay, so if we actually make a synthetic small regulatory RNA, which we target our target gene, and then we use this HFQ as a scaffold for that, then as in the microgravity, there is a decrease in the HFQ. So there is a signal difference, actually the expression of our target gene. That was our hypothesis. So to test that, so first what we do, that we want to see that what the E. coli, what we actually grown, if really the HFQ is reducing there. So these are a microgravity simulating reactor, which is developed by NASA. So what we do, use this thing. So how we do, so if the gravity vector is here, and this is the disk reactor, which is rotating. So this axis of the rotation and the gravity vector is perpendicular to each other, the cell or the particles inside the reactor is always in a free fall condition, so which actually mimics the microgravity. If you just put it horizontally, when the gravity vector and the axis of the rotation is parallel to each other, it's a gravity control. So first we test that the E. coli, what we want to use as a chassis for our synthetic gene circuit, we found there is a 50% difference in the HFQ. So we are now ready. Okay, so now we can use this E. coli to make synthetic gene circuits or a microgravity sensor. So here is our design, where our target is the EGFP. We develop some anti-EGFP synthetic small regulatory RNA, and which with the help of the HFQ binds with the EGFP mRNA and make it degrade. There is a silencing. So now we actually do the experiment in the microgravity and the gravity. You can see there is a around 30 times difference between the signal between the microgravity and the gravity. So this is the control experiment. There is no difference. So this suggests that this is the first microgravity biosensor we able to make where you can actually target your target gene. Now, one important aspect of this is not just the first microgravity biosensor, but at the very same time, this design, you can imagine that you can design this synthetic small regulatory RNA for any gene, which basically means that this design is universal. You can target any gene. So to do that, we actually changed this EGFP to the other target genes and the other fluorescence protein, and found it works. I'm not showing the results, but then we found, okay, why we give the target gene? Let us take some real inherent biochemical process and target that. So we choose the cell division process, and there is something protein called FTSZ. This protein helps the cell division process. So if you deregulate this particular protein, then you get a elongated cell instead of a divided cell. So you can see here, this is the yellow arrow. You can say these cells, which are in the Earth's gravity, using our genetic circuits, you can say we actually targeted this FTSG. We silence them. So you can see this is the elongated cells are here, right? Now, when we put these cells in the microgravity, because this goes through our design circuit, you can say these cells have actually rescued. So there is no elongated cell present. So in this way, we're very excited because we are able to show that using our microgravity responsive synthetic gene circuit, we can not only target any gene, but we can target a native biochemical processes through this microgravity. And this is just very recently accepted in biosensor and bioelectronics. And the summary of this this part is definitely for the first time, microgravity can be integrated as a physical signal with biochemical or cellular processes in a human designed way. And second is that the design of the device is universal. It can control the expression of any gene in E. coli with microgravity. And with that, I'd like to thank my group members. We have a small group, and the ANN work mostly done by Dipro and Kathakoli. The reversible computing work was done by Rajkamal, and all microgravity works done by Sayok. And this is the who found us. So I don't have a. So I just like to tell you that today the way I look at the synthetic biology and the conventional biology. So the conventional biology to me, what already exists, what we actually see, and what is the synthetic biology to me, that what can exist, which means the sky is probably the limit. We still don't know what is the design space of a biological system, but so the biology what already exists we do know through the evolution. Now to the engineering we can do, I mean, what can exist, and everything could be synthetic biology from the ground to sky. We all this, and thank you very much for your attention. Thank you.
Yeah, thank you, sir, for such a wonderful talk. I'm sure it will be helpful for all of us to know about the recent developments in ANN. Yeah, we'll now take up a few questions that we have received. Yeah, so there is one question. What does the activation function mean physically in the case of biological systems like bacteria?
Okay, so that's a very good question. It is not just for a biological system. So the concept of the ANN always comes through a mathematical abstract. So action this one. Okay, so basically it's a log sigma function. So question is how we are actually finding this nonlinear log sigma function through the synthetic gene circuit. So through the synthetic gene circuit, when the inducer is coming and your output is a particular protein, so they are not linear. Like if you're a biology student, you know there's the Michael-Menten equation where it is linear. So you give an enzyme and whatever the output you're getting is a linear fashion. So you increase that substrate concentration something, it goes to linear. But when it comes to a synthetic thing, when synthetic genetic circuits where it's based on the transcription and translation process, this is not linear, it is a non-linear. So how do we attain this linearity? This non-linearity attained by that is exactly here. So for example, if you have a gene expression process where before that gene expression starts, you need two molecules to dimerize. For example, two transcription factors to dimerize or trimerize or tetramerize before it. And this dimerization, if you do the mathematics, you'll see it gives some nonlinearity. If it is trimerized, the nonlinearity increases a little bit. If it is a tetramerized, the nonlinearity is more. So, but when we do the synthetic circuits, we actually change this nonlinearity, the degree of the nonlinearity, by changing the transcription factor interactions, their positions, how many of them are going to go there, in such a way. Okay, so I think I try to answer your question.
Yes, sir. There's one more question. Which mathematical approach you have taken and the work which you have shown as a part of mathematical modeling? There's this question in the chat box.
Okay, so yeah. So basically, as I said, so I haven't shown a lot of other examples what our lab is doing. So our lab also do like single cell. So our another goal is that how complex synthetic genetic circuits you actually can put in a single cell. So in level, what we use, which is the ordinary differential equation. So, and then you may have a very big equation to actually do that. But when we come to the ANN, we are using the same mathematical formula what ANN people use. Either you use in the Facebook or in a mathematical biology paper. So ANN has a very fundamental mathematical framework. So, and that mathematical framework is, as I said, so there's a two function. Or maybe I'll go here to give here. So here you can see all inputs are coming. They're connected with the multiplied by their weight function. Then the summation is that x1 w1 plus x2 w2, and then there is a something called a bias function. So this is the linearly you combine that, and then it goes through this function. Whatever the output from this summation is go through a nonlinear function, which is log sigma function in this case. So that's why the mathematical framework from ANN is easy. Now, what happens in when we have a big ANN network, in this ANN network, the way you actually change the W values, because W values can be from anything from to minus infinity to plus infinity. So you can actually permutate all these values. So they have a different kind of mathematical techniques to how we actually change to the next W values. But in reality, if you randomly change, you can also come to that moment. So in biology, but in biology, how we are changing this particular W values, not in the mathematics, but actually in the molecular engineering. They say changing the transcription factors interaction and something, and that changed the W. And when we have an input-output relationship between the input and the output, we actually again we fit this equation, this log segment equation with this data, and then try to find out that what is our new W is. In this way, we are actually making this W. I just I'm maybe taking a little bit time, but I just want to tell you one thing. Maybe I'm not clear at one moment. So, for example, I give one Facebook example. Okay, in the Facebook, if you want to open the Facebook in your mobile phone, and then there is a face recognition, don't believe that your mobile phone is so good that this the change of the W's and all this all the adjustment is happening in your phone. No, it is not happening in your phone. It is happening in the Facebook server. And then when the final weight adjusted neural network is developed, then you actually plug in directly to your mobile, right? So similar thing we are doing here. So here what we are doing, we are our server is our lab. In this lab, we are changing these molecular interactions manually, and then when it is done, we are actually upload on the microscope. We're seeing that. So this is the connections between the how people do in the Facebook and how biologically we do. Thank you for your patience.
Yes, thank you, sir. So I thank all the participants for attending this session. Continuing this, we now request all the attendees to shift to the Promega Hall for the next event on Microsoft Teams. The link for the Promega Hall has been posted in the chat box. Thank you all and see you at the Promega Hall. Yeah, thank you.
Thank you.
Sir, excuse me, there is one more question in the chat box.
Yeah,
which
sure.
Yeah, which mathematical approach you have taken? Yeah, okay. Yeah, no, that's it. This is message. Thank you, message.
Okay. Thank you.
Yeah. Thank you, sir.
Thank you.